what I'll do currently for us is that we'll go through some of the, essentially the sequencing and some of Illumina's core technology and how our biochemistry works behind essentially our amplification strategy and our sequencing technologies. So we can discuss this as much detail as we want. So the workflow. Last stage of the ultimately double-stranded DNA library that's been created from the samples. They all have indexes on them, and after PCR, they should all be. And that's you know, the 2-day prep. That's very standard. And we will look at essentially what happens next. So quantifying things and qualitatively visualizing them is very important because essentially we're going to go into cluster generation. So Illumina is a chip company. We made microarrays for many, many years, and we make chips. And our sequencing platform also runs off a chip. The chip is pictured right here in the top of that is what we call a flow cell. The flow cell is just another rendition of one of our chips. It is where all the reaction, the amplification, and the sequencing occurs, particularly in the NextSeq that all occurs on the instrument as well. So you can see here, this one is it's actually a high-seq flow cell. There are 8 separate partitions that are sort of located here, separated by these little lane dividers. These are actual interlanes for the flow cell. Your NextSeq, as we saw from the data before, has only 4 lanes. But the lanes are much, much bigger. So you actually get a lot more reads and things like that. That's a lot to do with the hardware. And what's ultimately pictured below it is our P5 and P7 grafting primers. Those are the primers that are complementary to the adapters that we've just ligated onto our libraries. So what I'll do now is I'll kind of go through this cartoon, which sort of depicts how the clusters are formed, how you deposit the library onto the flow cell, and how the bridge amplification works. So lanes are good because lanes allow you, at least on this high-seq platform, to separate samples and things like that. So if you can separate samples, you can actually use the same indexes and things in each lane. Lane gives you a physical partition. For the NextSeq, you will not be able to do that. It relies heavily on only the indexes. So just be aware of that. So, but in any case, how do we make clusters? So ultimately, a single DNA library, and this sort of encompasses a lot of platforms. So I apologize, it's not necessarily specific to the NextSeq, but essentially our cluster generation takes a single DNA molecule and goes through a amplification strategy which creates a sort of a very localized physical space of other molecules, and this is the clonal amplification that we do. That localized sort of cluster of molecules is what we term a cluster, right? It's a sort of a physical isolated entity of this single molecule that gets amplified, if that makes sense. I will show you. In some of the platforms, there's a sort of extragenous piece of equipment we call the CBOT, that is the clustering bot, which actually drives the clustering and things like that. Some of our higher throughput platforms require that, but the NextSeq does not. So the NextSeq is sort of pictured over here, where you can actually do the clustering and the sequencing all on the same instrument, which our sequencer does both. So once you have this sort of single DNA library, and again, it goes through the amplification, and this sort of physical aspect that's being represented by this little circle here at the right here is essentially your amplified clonal cluster. So, oh, I can.
That's the part that's coming.
So cluster generation.
Yeah.
Cluster generation does. I think there's essentially 4 steps in it. And I'll step my way right through these, and you'll see the steps sort of highlighted up here. So step 1 is essentially to hybridize your fragment and extend. So what's depicted here, you can see, and I love the shadows on the oligos that are grafted across the flow cell. That's what marketing does, right? They can do things like that. That's right. So these are your P5 and P7 grafting primers that are sort of distributed all the way across the flow cell. The flow cell I can mention that if you see it, you will see some when you go over to Woodbury. There are thousands of them there, literally. All different kinds too, MySeqs, HiSeqs, NextSeqs. You'll be able to see that it's essentially two pieces of glass, which just has a polymer that's in between those two. And whether you have 8 physical separations or not, it doesn't matter. You have 2 here, the MySeq only has 1, the NextSeq has 4, whatever. All the principle's the same. It's glass, it's like acrylamide, and it's glass. And that's the acrylamide is what we actually deposit all the clusters on. So you can see that it's a solid phase, right? They're all sort of bound to this sort of polymer that's in between these two glass. And the lane itself is just an extension of the fluidics. It's what drives the reagent delivery and things like that. So imagine it as such. A lane is simply the tube. It's just an extension of the tube that goes through your samples. So and the flow cell facilitates that. But the first thing you do when you do cluster generation is you have your blue library fragment that's depicted here, right? And the adapters are obviously on the end. You do the hybridization to, I think it's P5 first. I will mention that this is a single strand at this point. Its counterpart, its antisense, is also in the reaction as well. So one of the first things we do before we do cluster generation, as you know, we have a double-stranded DNA library here. We denature those strands with sodium hydroxide and we just split them apart. Each strand gets deposited as a single strand onto the flow cell. And then it goes through hybridization. And once it hybridizes, and the machine facilitates all this with temperature and things like that, right? We decrease the temperature to hybridization, we increase it to 55 degrees, and we do an extension. And essentially, that extension is, the first strand is done with a very high fidelity polymerase. That is to alleviate any errors that you make in that, essentially, that first copy of that library molecule. And the reason we really want that to be error-free is because this is the molecule that's ultimately going to drive the bridge amplification. The library that you just spent 2 days making, you're going to flow some more sodium hydroxide, you're going to get rid of that library. That library's gone. And all you're left with is a copy of that library. So that copy, however, because it's been extended from one of the covalently bound primers, is now stuck to the flow cell. So if you look at things this way, you have a single strand of DNA, and what I have over here is essentially a very poor image of what cluster density is sort of looking like, right? So each little blue dot that you have on here is essentially representing a sample that's been clonally amplified. And the correlation that I'm trying to make is essentially the delivery of this template in this first extension is what drives how many reads you're going to get and ultimately cluster density. So if you put too many these molecules down, you have many, many, many dots that are forming here. And what I have highlighted is essentially, when a cluster is in a sequencing reaction, it's far away from all the other clusters, it has good background, it has good signal to noise, it performs very well. But the more clusters and more template you add, you increase the chance of getting guys that are very close to each other. And those are some of the things that pass filter, or sorry, that fail filter, is because they're too close. They can't be determined to be a singleton, and this is all sort of related to density, right? And why the quantitation and everything is going to be so important today. Because if you don't know how many molecules you're starting with, you don't know how many molecules you're putting on a flow cell. And if you put too many, it's a disaster. So, and if you put too few, you limit your yield, right? And you're not going to get to 400 million reads. If you only load half, you're going to get 200 million reads, right? So that's cost ineffective. So having that understanding of a fully functional molecule and how many there are is extremely important to operating the platform successfully and optimally, right? So that's why we use qPCR. We use qPCR because we literally determine how many molecules are functional in solution that have both adapter ends and can amplify. So if it amplify in solution, you can amplify in the flow cell, give or take. In a solid phase is a little harder than an aqueous phase, but nonetheless, the chemistry has been worked out to the point where it actually grows very well. So,
I have a question.
Mona.
Aren't the primers fixed to the flow cell?
They are covalently attached to that acrylamide layer, yes.
Right.
It's all
so
that.
I never understood, why don't they make sure that they're spaced far enough apart so that you never can get an overlap in cluster density?
Welcome to Illumina's new machine called the X10,
Oh my God.
which it leverages what we call the ordered array. So we're a chip company. So here, the primers are randomly associated across the flow cell, and you're really driving density by dilution, right? You're adding the right amount. So you're either diluting it till you get, that 230,000 molecules per millimeter squared. However, on the X10 platform, we have an ordered array, so things are not randomly distributed, and you follow very similar Poisson distributions and things like that. You get blank spaces and double space, and all that. The ordered array, there is, if I remember this right, 2,354 pads. We call them pads. They are essentially divots in the glass, and they are extremely ordered. And they are 700 nanometers apart from each other. That way they don't bleed into each other, and you can ultimately fill a flow cell with a whole lot more clusters when you do that, because you know where they're at. That does some other analysis impacts and things like that, because doing things randomly, you have to find clusters and build clusters and build the intensities and the sequences and things. Ordered arrays, well, I know where they're all at. They're all right where I made the order. So you don't have to do any of that analytical stuff either. You can just go through and pick the spots and call your intensities. So, but that's a different topic. That is what an ordered array is. So.
Technical challenge to develop that.
Oh, it took us many, many years to get that sorted out. Trust me.
That's previously it's just sort of random by dilution.
Yes.
You're gonna lose.
And that's what we've been leveraging since then. Yes. So, the products will continue to evolve. So. Right now, the ordered array is only for the HiSeq X platform, but again, there's the evolution. Illumina won't make 2 flow cells if it doesn't have to, because that takes a lot of time and effort, right, and a lot of money. So if we can make 1 flow cell for all the platforms, we will. So
he didn't ask me to ask that question to introduce his new Illumina stuff, I promise.
the X10 doesn't have anything. Unless you're really interested in sequencing thousands of people in whole human genomes, the X10's not for you. Literally 18,000 a year you'd have to do. So, and you have $10 million too. So that might be another limiting factor. So once you do that extension, right? The marketing draws these slides so that the strand is sticking up in the air, right? That doesn't happen. That's not the way it works, right? It's a single strand of DNA molecule. It's like this big ball of mess that is tangled around everybody. But we do the essentially the regulate temperature, and we flow some. Denaturing its formamide to sort of disrupt some hydrogen bonding and facilitate the annealing and stuff. But the other end of the molecule is actually the reverse complement of your other primer sequence, the other adapter, right? Because you've extended that. So you've made P5 now P5 prime. P5 prime comes over and hybridizes the P5. And this is what we essentially what drives what we call bridge amplification. So again, a polymerase comes in, and you just do your bridge and you extend it. Now you've got 2 molecules. So. You can kind of see where this is going, right? And then you cool things down again, and everybody bends over. And then you got 4 molecules in. We do that 35 times. And we do that over and over and over and over again so that you get a very localized, essentially high-density lawn of single molecules that have essentially been clonally amplified from one starting library molecule. And you have all this. And they all look like that, but that is one independent spot, one independent cluster that you see. It's one independent read that'll come out. So there are, on the NextSeq, there are 5,000 molecules per cluster, give or take. So we roughly estimate that at. Other question?
Yeah, but then the molecule in that cluster represent the 2 complementary strands.
Yeah, absolutely it does. Okay. You are on the ball, I'll tell you what. So. You're right. One is going one way, one is going the other way, and they are complementary to each other, and they do touch each other and anneal to each other, and they do cause a lot of problems. So what do? We get rid of one of them, and we get rid of one of them through the next process called linearization. So if you so the first one was essentially the extension and the amplification, right? Once you amplify things, you're not done. There's 2 more steps you have to do. 3, sorry. The first one is linearization. So if you look, it's not so clear here. But there's on this grafting primer down here, let's say it's P7, there's a little green dot. It's darker green than all the other green dots. That is there to signify a modified base. So there's only a couple modified bases, and that one's a uracil. So what do we do in linearization? We flow in glycosylase. And what does that do? It comes in and finds that uracil and cuts that base off. It liberates that strand from the flow cell. So now, you only have 1 strand, and they're all facing the right direction. The other strand has another modified base. It's guanosine modification, which uses user enzyme or something like that. I don't remember fully. But in paired end reads, you just toggle between those 2. Does that make sense? Linearization gets rid of your.
The other strand is a modified. I'm sorry, I got the U glycosylase, got that. The other strand is
Oxy-G guanosine. I don't remember.
some modified G, but is it
Yes.
glycosylase also? Or?
No, it is user. User enzyme, USER. I think Annie B sells it. I don't remember.
So that's the way you prepare the molecule before they
Go into sequencing.
filter it onto the flow cell, right?
This is happening while it's on the flow cell.
Wait, can you just say again? You called it user. What was the name of the enzyme?
User.
USER, so
Yep.
I can look it up? Okay.
Yep.
And it's NEB?
I think so.
Okay.
Don't quote me on that, I think so. I don't remember so much.
So that law, that U, is that already?
It is grafted onto the, it's on the primer on the flow cell. That is right, that's how we make that. So all of that chemistry is incorporated into the flow cell manufacturing. And then the other primer just has the guanosine modified base on it.
Then you need to do paired and sequencing. You already just got rid of one of them, right?
We did.
So how are you going to read from another side?
We're going to bring it back and read 2 turnaround. Oh, yeah. Essentially does another round of cluster generation. So, yes, those are all very good questions. So once we've liberated this strand, right, we have everybody that's kind of facing the same direction. It's all the same molecule. We do one little thing is we block with a dideoxy. So all open 3' ends, right? Because again, they're single-stranded molecules. They're flapping around, and this end here can easily hybridize somewhere else. And then you start an incorporation, right? And all you need is a primer site, and the polymerase will incorporate a base. So we block all that. That greatly diminishes the noise that we see in our sequencing reaction.
So you just add like a single nucleotide?
That's absolutely right. Terminal transfer state. We'll stick that dideoxy on the end of that. Yeah, I think so. Either that, it's tack polymerase or
Is
something.
it terminal transferase or
I think so. I don't know.
terminal transferase like?
Yeah, it's sticking that dideoxy on the end. To be honest with you, the formulation of things change a lot. So I would say at one point we used terminal transferase. I can't quote if we're using that right now or not. I don't remember. But anyway, yeah, we block everything so to prevent any like spurious priming sites and things like that. So strands are all facing one way. They've been cut. They've been blocked. And then the final step is to just slap a 20 base sequencing primer on this thing, right? And you hybridize that gives you a priming site, and you are ultimately done with cluster generation and ready for sequencing. On the next seq, this takes about 2.5 hours to do that. Sequencing primer. So I'll talk a little bit about sequencing, then I'll go back to read 2, because that's the way the machine operates. All right, sorry.
So how did you get the other strand? I mean, do you do 2 clusters for
We
each
have,
strand?
we're not there yet.
Okay, sorry. Okay, I'm sorry. No.
No, it's okay.
That's all you do, like, oh, I got it.
Yep.
I missed it.
So we do,
Okay.
in sequencing, and especially paired end sequencing, they're consecutively done. So they're not read simultaneously. Sanger you read from both ends, right? That's not the way it does on the Illumina's platform.
No,
Yeah.
it's fine. I just want to make sure it's fine. That's totally
Sure.
just want to make sure I missed it.
So things have been amplified, things have been blocked and primed and linearized. It's ready to go. We go into sequencing. Sequencing is very, very simple. So this blue strand is essentially just a magnification of what we saw before, right? We have our block, we have our primer. So we use an SPS. It's sequencing by synthesis. It is essentially our IP on this is the reverse termination fluorophores and blocks. So I'll show you that in one moment. But what this little cartoon is trying to show you here is we flow all 4 bases at the same time. And I won't come into fluorometry so much, but the sequencers themselves, they're just fluorescent microscopy. That's all this is. It has an objective, it has a camera, it has an excitation source, and then you have a fluidic system which actually delivers the reagents to it, incorporates the base. It's a very simple platform. But ultimately what this is showing you is that we flow all 4 bases, and due to hydrogen bonding, the polymerase that's with the nucleotides, we'll consider the nucleotides to be 4 different colors at this point. Sometimes that changes depending on the platform. But the 4 bases come in, they have all 4 nucleotides, one gets incorporated, we wash all the other ones away, we take the excitation source, and we fluoresce the fluorophore, we take a picture. Then we use our reversible termination, or am I? To both cut that fluorophore off, and I'll show you sort of a generic picture of a nucleotide next, but in correlation with the fluorophore, there's a 5' hydroxy block on it. So you can only incorporate 1 base at one time. You can't incorporate any more. So it's just 1. So every cycle that you do, every incorporation you do is a base in your read. So if you want a 150 base pair read, you are going to do 150 base pair cycles. Oh, sorry, 150 cycles. That make sense? Do 1 at a time. You step your way down the strand just by incorporating bases and taking pictures of what fluorophore is on that base. That's all the sequencer does.
You're analyzing the image to find the clusters and
Yeah.
which colors they are.
Exactly. We look at pixel space. We start at 0, 0, and we mark down all where the intensities are found in the entire pixel space. And then we go through and we lead the intensities that are from that. And what intensity it is, it's pretty much the highest intensity we see, we call. So you have all 4 in there. It's A base. It's highest intensity. You find it's an A base.
So is the cluster size pretty equal, so the intensities are pretty equal?
It is, and that will be actually related to insert length. So the bigger, you can imagine, the longer the insert fragment you have, the further the bridge is actually able to go for another primer. So what I see, and what we see in the community is, if you have things that are 500, 600, 800, well, I'd say 800 bases in length, and you adapt it, right? You have like about 1,000 base pair molecule. That'll actually create a much more less dense and dispersed cluster. So you lower signal to noise and things like that. So that's why there's a lot of restrictions on insert sizes. That's why we keep things small. And the library prep that you're doing is keeping everything about the same size. So your clusters are actually all the same size. If you start mixing 800 base pair fragments with 100 base pair fragments, the software will have a hard time determining which cluster is which, just because there's a diversity, and the guy doesn't like that so much. Right? He likes things to be very uniform and things like that. If you start to variability and deviation, do bugs out a lot, and it'll start calling wrong clusters, and you'll see drops in quality and stuff like that because of that.
I know it's proprietary. I mean, I don't know how much it is proprietary, but can you talk a little bit about what the block is and
Yeah.
how it
Yeah, sure.
how I see something? There's a miracle.
And so the machine has, yeah, it's pretty easy. So you get the idea, right? You cut the fluorophore block off, and you just do this again and again and again and again, right? And you just work your way down the strand. And then that gets base called. So here's what our nucleotides look like. Obviously on here on the left is prior to blocking or deblocking. But each one has a separate fluorophore, so consider 4 colors in here. But there's also this 3' block. I think the block's just a phosphate bond, to be honest with you. Right? It just keeps things from extending anymore. And we cut that off by using a phosphatase, right? Give you a free hydroxy group, and away you go. The cleavage site here, so cleavage has changed a little bit. In the sequencing reactions, there's essentially 3 active components. The incorporation mix, which has the nucleotides and polymerase. And then we have the scan mix, which is essentially just sort of a dye protector that we flow over things that when we do the imaging and things. And then there's the cleavage mix, and the cleavage mix is essentially, it's all in one. When it comes in, it cuts both the fluorophore off and liberates this block and deblocks things. And then you wash kind of all that away, and you're left with sort of a modified nucleotide here with no fluorophore and another hydroxy group just to incorporate the next base. So I'm not 100% sure what this is, but we can mix and match components and things like that. I think they're chemical cleavages. I don't think they're enzyme. So maybe the phosphatase is, but I'm not 100%. It is extremely efficient, so that tells me it's oftentimes a chemical cleavage. So it's like you can use a sulfur bond or something like that, and we're just oxidizing that to cut that bond, something like that. That would be the principle behind it. So.
So that means that, for example, if you have a row of G's, there's a good chance that you're going to have a bleed from the first G to the second G, and that's it.
Not necessarily, yes. We don't suffer those, I guess, multiple the homopolymer runs and things like that. That is right. We had a tremendous amount of problems with those due to the essentially the system and the way that was done. I know very well that platform, so that a lot of those things actually come out of the well, right? And you can't capture them all and they go floating away. That's the problem with that platform. Here we do not, because we do one. So if you have a stretch of 100 A's, you will get 100 A's, pretty much. Yeah, sorry, I lost my place here. So you see the idea behind incorporating the bases and deblocking. That is our reversible termination chemistry. That is what we've essentially have IP on and what we've built our sequencing technology behind. And all of our platforms do exactly the same thing, give or take, pretty much.
Where do sequencing errors come from? From the polymerase not being very...
Yeah, so our errors are they're pretty minimal, but they do encounter something. So we see more N's than we do errors. So indels and things like that we virtually see nothing of, right? There's very seldom any case where you would miss parts of the read and things like the insert and stuff like that. That doesn't. With some of the homopolymer problems, you do incorporate those, right? Because you get 10 instead of 8 A's or something. So your indels will show up there. We don't have that issue because of the Terminators. But we do have SNPs, and that is ultimately something from maybe too much bleed. So the system is very fluidic based. So ends come in a lot of times because of bubbles and things like that. So a bubble will come in and it'll essentially obscure the image to where you can't see the clusters behind it. And if the analysis software doesn't know what that is, it just makes it an N. So it'll just give you an ambiguous base, right, at that point.
You just get the bubbles
Because it's sucking things through a manifold, yeah, that's all. It's, you can't beat the bubbles pretty much, so. They're inherent to the platform, so. But SNPs and things like that, just too much optical bleed maybe. You know, you get little specks of things that fluoresce oftentimes. I would think in most SNPs, most incorrect base calls, I would like to see that SNP or that incorrect base call associated with what quality score that is, because I bet that quality score is lower than all the other ones. Things like that, so we correlate those together. If you have a wrong base call, we can go back to the quality. I can assure you in the Illumina platforms, that quality is going to be lower. Things like that, because it will affect other things. But again, the polymerase is just incorporating something, right? So, and taking an image, your SNPs and things will come much closer to the end of the read. So your signal, it's all about signal to noise in these platforms. It's all about intensity. If you got good intensity, you got low background, you're going to have awesome base quality. As things kind of progress through the run, you start to get the droop, right? It's an enzymatic reaction, it is. And the polymerase starts to die off, the fluorophores don't fluoresce as well, you start to bring yourself closer to signal to noise, that's where you get some missed calls sometimes. So I would expect them, a lot of them would be associated to the very end of the read, things like that. You can trim that stuff off. Things like that happen, that's where they come from.
What sort of polymerase do you use?
Ooh, at this point, yeah, so it's a strand displacing, very process oriented polymerase. So it doesn't let go, it doesn't open and close like go. The one we use now, so Illumina bought Epicenter a long time ago. And most of their enzyme is what is incorporated into this platform. So that's the reason we bought them.
So it's an E. coli polymerase?
I don't think it's eco or one. Honestly, I have no idea what it is. But it'll be some. It operates at 55 degrees, so
It's a regular.
it is some sort of thermophile coming from. It's an archaeobacteria polymerase or something like that. To be honest with you, that changes a lot too. So.
And is it, because I see this, because I was wondering why would it have highly precise, oh, because you want to stay there in between rounds.
Mm-hmm.
Got it, okay.
You want it.
Actually have to have high fidelity, less the time.
So fidelity is not as important
Just have one.
after the strand because essentially you have 1,000 molecules, right? And in order to see an artificial error, you'd have to have all 1,000 of those molecules do the same thing, and they don't, right? It's a fraction of a percent. Maybe 1 out of 1,000 may miss an incorporation or something. It'll fall behind. We have terminology for that. It's called phasing and pre-phasing. So there is certain shifts in phase where things are. Everything in a cluster should be in the same phase, right? They should be, all molecules are going boom, 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 but sometimes one does too. Maybe a block didn't happen so well. In synthesis of the nucleotide, right? Those inefficiencies happen in the molecular chemistry. Maybe the polymerase doesn't incorporate a base, right? That guy falls behind. So he's always one base behind, right? So you start to get out of phase, and again, the further out in the run you go, the more you'll start to see that, right? So that's
More
what,
of them would be
a cumulative effect, absolutely, they'll add to that. So that's what limits a lot of our read lengths, right? Or we'd read thousands of bases if we didn't have that problem. So we stop things at 150 bases because we know after 150, the phasing gets too dodgy to accurately call the base quality, things like that. All of that's sort of embedded into the system at this point.
What about the amplification step? You must need a high fidelity.
Yes, that has been mutated pretty well too, I think, to where it's able to do the bridge on the glass and things like that. Glass is very positively charged. It causes a lot of problems, things sometimes. So yes, we an entire group of people that did work on that. Yes, and what I have here is obviously the images are not color coded quite so nicely as this. They're merely black and white, right? And we use filters and intensities, which essentially base calls everything. It's all visible light that we see, so it's all white. The images you see, I think some maniac in marketing went through and actually colored all these dots in here, so that shows you some sort of representation of what's going on in the general scheme of things.
It's actually
Yes, he's copying the same thing over and over again, right? But anyway, it's giving you a visualization of how the machine is looking at things, right? So it's looking at a specific spot and seeing whether it's blue or green or red, it's like that. It's kind of what this image is trying to.
Does the machine basically then remember where a spot is?
Oh yes.
Next run, look for what the color is at that same position?
That's right. So that's what registration does. So registration does 5 cycles. So it essentially looks at the first 5 cycles, and it determines what pixels contain an intensity. and what pixels do not. And it marks those that do not as background and those that do with intensity. And then it records that value. It's recorded in a file called a location file, lock file, LOC. So that lock file gets carried through. You only do it in read 1, because read 2, it's the same location. You just resynthesize a strand. I'll show you that in a moment. It only does it in read 1, and it picks that spot, and it just calls intensities throughout the entire cycles of all the cycles that you do in that run. So that's the idea behind registration.
So is each, I think I missed something you just said, each nucleotide have a different color?
Mm-hmm.
Okay, so it's got a different color, but you keep referring to intensity, so I'm confused. about
Intensity-
color versus intensity.
Intensity is the color I guess is the color from the specific channel. Yeah.
Amount
Yes,
of
is the amount of fluorescence from that channel or
So
from that
if-
fluorophore.
If you've done the same number of cycles of each strand, and if your strands are a comparable length to start with,
Sure.
shouldn't each cluster be?
They'll be normalized, absolutely. A lot of our intensities, I can show you the raw data and things like that that'll show you called intensities. It goes through background subtraction,
So does it?
a local
so
background
essentially
subtraction.
So I understand that the color is telling you which nucleotide is
Mhm.
there. The intensity is telling you
That it's there, that base has been incorporated. That's all you need to know. You don't need any quantitative value on that because you're only doing one at a time.
Well, you're just calling it 1 and 0.
Oh, you're
right?
only doing.
Yes, it is. It is
Right.
literally
You
going
don't
tick,
do
tick,
all
tick.
four bases at once with
Nope.
a different color. You do
Well,
one.
for the
no, no, no,
registration,
no.
just for the registration.
Oh,
The registration is
But-
there a cluster at this position, is the question. So-
Literally, yes,
Okay.
yes.
I see.
And it doesn't care which color it is, right? That's why it looks at 5
I
cycles,
see.
right?
I got it.
Because, if
I got it. The
it helps
intensity-
differentiate things, yes.
Okay.
A lot of this will come out in the data analysis, which I won't touch too much on. But yes, that is a whole different world of how that goes through and does things. The online material for that, some training videos and things like that, check them out. They'll go through all of the pretty much what we call RTA, real-time analysis. That's the program, that's the software that's running behind the instrument and doing all of your image extraction, your intensity extraction, your normalization, your base calling. A little bit about data analysis. There are essentially 3 spots here I'll just briefly mention. There's a primary data analysis, and I may tell you something here, it'll probably confuse you more, but it's okay. Secondary analysis is obviously some of your alignment and variant calls and things like that. And you have the IGV from Broad and things for data visualization, depending on what you're looking for, right? So some sort of GUI-driven visualizing tool. So just to touch upon things, sequencing-wise, I mentioned RTA, that's a real-time analysis. That's what's doing the image extraction. There's also the ICS, that's the instrument control software. So that's ultimately what's driving the instrument and flowing the reagents and taking the pictures. Primary analysis then, essentially this is generating your TIFF file. So if you think about how the machine works, right, and how it runs, it essentially generates three sets of outputs, right? So the first thing it's doing is taking images, right? It's taking images of the bases that are incorporated, right? So it ultimately takes 4 images. It takes 1 for each color channel. And whether you have a fluorescence in that channel, it overlays the images and it calls the highest fluorescence. That's how it does that. And then you sort of get all these little TIFF images and location files and things like that. Once you have the images, you can obviously find the intensities per spot. So the intensities get spit out here in the primary analysis, and that's really what comes out of RTA is essentially an INI file, an intensity file. The intensity files, like I said, RTA goes through and there's some filtering that goes on afterwards, but at this point, it just calls the highest intensity the whole way through the read, and that is what it is. And then some of the quality scores and the predictors will come in afterwards, right? So you can think about if you have 2 intensities in a given cycle that are very equal, some of the filtering will come in. Instead of calling that a SNP, it'll just either discard that read if there's too many of them, or end that base if it doesn't. If there's some ambiguity on it, it doesn't call anything. So.
What if you have a heterozygous situation?
Oh, it'll call you. Yeah, so one went here to the cluster, one went over here to the cluster, right? We split those strands right at the beginning, right? So you have 2 different, you'll have 2 different populations and things, right? So, yeah, sure. They will not be together in one cluster. But when you do all the reads, you do all the alignments, you're going to see 2 populations, 1 from each chromosome, right? And obviously secondary analysis, Illumina has some software themselves and base space and Cassava and things like that. We will be using the subway for at least this course in any case. And that'll do your alignments and variant detections and things like that. So I will briefly highlight the read 2 thing, because so the read 2 is very similar to what we just looked at in read 1. And essentially read 2 is doing another cluster generation. So it's taking the molecule that you have, and unfortunately this doesn't do the greatest job in picturing that, but the molecule that you've ended with in read 1 essentially gets deblocked, because there's that block on there. If you cut that block off, you just reform the bridge and you re-amplify read 2. And that re-amplifies the other strand, and you come in with a different linearization enzyme, cut the first strand that you've sequenced, hybridize the second strand, and read the second strand. So the key of paired end sequencing is that cluster is reformed at this exact same location where read 1 is. So you know where it's at already. You know that read 1 starts at the 5' end, and read 2 obviously starts the whole way on the 3' end. So you have both ends that you can sequence, and that gives you sort of the paired end sequencing strategy for things.
Because the clusters are
It just
attached to
re-rows over. Yeah, I don't have it so much here, but that's the idea.
Same thing, it's gonna get to one of the oligos still bound on the plate.
That is right. So that oligo is still there.
It's all like glycosylated away?
Only half of them. The other ones are the user. The modified base, depending on which primer. So P5 has the glycosylase, and P7 has the guanosine. That way you have one on one side and one on the other.
So there's gonna still be some P5s on the cluster that,
No, not necessarily.
okay, because
Can you go to that one picture where you had them all, like, where you explained what the first one was?
You know, let me see something here. I have something else, I think.
it seems like, because your P7 one is attached and you just sequenced it. But now it's gotta make a new cluster, it's gotta bind to a P5 one. It seems like you cut all the P5.
But that is P5. At the end of P5, they have the two, right? So they cut it on top of P5. So P5 is still there, right? So they just cut everything on top of
Yeah.
P5.
So you have a little chunk there. Yeah.
Got it.
Right, so they can still
Okay.
use
So that linearization base is actually 20 bases up the oligo, right? So you cut that last base, that's okay. You got 19 left, and that's ultimately what facilitates. So this presentation may be a little better for us, right? Primer, and you do the sequencing, and then is it in here? Paired end sequencing. So here's where I'll do this for you. This will make it clear. This is where we stopped off in read 1, right? So you have your primer, and then you have essentially, this looks like a 25 base pair read or something, right? You've done like 25 cycles or something that you've incorporated there. So step 1 in paired end is deblock everybody, is take that block off. Whatever that may be, the dideoxy or something, somehow we get that off of there. I'm not sure how that goes. But then essentially you just cool down the temperature and you can re-anneal to the other side again, right? So you come back and bind to that piece of primer that's there, give or take a base that you've cut off, and then you do an extension. And essentially you've brought back that strand that you got rid of in the first place. And then you go through and you do, essentially on paired end, it's only 15 cycles. Do we know why it's only 15 cycles instead of 35 in read 1? But you don't start with a single molecule in read 2. You already have 1,000 molecules. So you do less cycles because it's a little more efficient, right? 35. And now it's 15 for read 2. So yes, essentially it does this, and you bend that over 15 times, and then come in with the different linearization enzyme, cut the other strand off that you've already sequenced, and then sequence your read 2.
That's the one that has the 45 genes.
Yes, the other one, yep. Block the ends, and you're ready to go for sequencing. The same way you did read 1. It just kind of turns seq. So the next seq, it only has one set of cluster reagents. It literally uses the same cluster generation reagents for read 1 as it does for read 2 turnaround. It's identical in chemistry. So it's all the same. We only have to use the same thing. So, and then I have the indexing thing. So indexing, I wanted just to make a comment. Oh, sorry.
So is that routine to read it twice? Or
Yes,
is it okay?
I would think everybody. It depends on the application. All right?
So
We'll think of it that way.
So it seemed like you said that a lot of the problems that you identified that you might have with the reads were as you got to the end. So this might be a way of helping.
Oh, that's absolutely a great idea. If you can, so what you have the ability to do, a lot of people do, is you make a library that has a 100 base insert size, and then you do a 100 base sequencing from both ends. That way you cover the read 1, covers the entire 100 bases in this direction. You turn around and start read 2, you cover the entire 100 bases in that direction. You have a completely complementary overlap of reads. So any directionality-induced problems will go away with paired end sequencing. It builds your confidence, right? That's all part of the quality scores and predictors and things that go into that. It's a very good idea to do. Some people only overlap 25 bases in the middle. That way they can do the entire length. Yeah, so the MySeq, you can do 300 by 300. If you overlap, 50 or 25 bases in the middle, the software actually has the ability to stitch those reads together, give you a 600 base pair read at the end, starting from one to the other. Very useful in amplicon sequencing. The MySeq generates about 25 million reads per run. So when you have a very focused target and amplicons looking for low limit of detection, that's the platform, that's the way you want to do it. That's what people use that for. So.
How many base pairs do you typically read then? 150, is that what you said?
So the next seq, the max is 150. But you can, as long as you have 25 bases, you can align things pretty reliably, depending on mouse and things may have to go a little higher, just due to redundancy and things like that. But you're looking for a strength, essentially a length of a read that'll give you a specific alignment, right? It's something that'll, it's not ambiguous in lining in multiple places, right? So if you got to go further, you go further. So, and again, length is also increasing your coverage and your depth of across things. So. That's all good fundamental things to understand, right, when you're sort of designing your sequencing reaction and things like that. Reads, I will just mention the index stuff. We'll introduce more reads. So there's not only a read 1 and read 2. Technically, for our library, there's going to be 4 reads. It's going to be a read 1, an index 1, which is actually read 2, an index 2, which is actually read 3, and a read 2, which actually is read 4. So if it didn't get any more confusing, there you go. I know, it's horrible. So yes, I'll go through it very slowly. So what this is kind of trying to show you, we even though we consider the technology read 1 and read 2, the way the machine runs, it numbers the reads in consecutive order. So when you do a single index, which is what the data that we got off Cold Spring Harbor recently did, we do read 1, we hybridize the primer, we read 150 bases or something like that. That primer gets dissociated, gets denatured off. We just put another index primer on here, which is actually reverse complement to the adapter on the other side. In between our 2 adapter sites, there's the barcode. And depending on which barcode we pick from the...
Make it all the way
Yeah. So
Let's make it all the way down.
this little green thing right here, our indexes are only about 6 bases, 6 to 8 bases. So it goes through and it just bangs out 6 bases. They're a unique identifier. That is the 6-base read, which is actually going to go into demultiplexing and split all of your samples apart from each other.
I thought you would index these on each side.
We do.
Like they're actually string.
When we do dual indexing, we have 4 reads. So there's 2 index reads. They occur right in the middle. So you have read 1, which does read 1, and then you have the second read, which does your index 1. And then you denature that. But things are facing the other way. So you can't necessarily hybridize we're looking at 3 to 5, right? So you can't hybridize anything into your insert. So we actually re-anneal this down here. We do a couple of base insertions, something we call dark cycling, where we just insert bases witho't taking images. That gets us to the primer site and essentially reads the second index, which is lying right here on the 5' end of that second adapter. And then once we do our index, we do the chemistry, we turn read 2 around, we put the other primer on, and away you go. So your read 2 is technically your read 4. That's where that comes into. And it's simply because we label the reads in consecutive order.
So what I don't understand why you actually have is you do the index read because you're not sure that the sequence in read 1 is going to go far enough to actually read
No.
the index. Is that the reason?
You do the index read to separate all the samples that are mixed in the tube when you put it on the machine.
No, I know, I
All right.
understand that, but my understanding before
Oh,
is that
you will not
that
reach
would
the
be
index.
part of the in sequence that you get from read 1.
No, your insert is much longer than your essentially your read length.
Okay.
So the first thing you ought to do is there's an adapter sequence that's in front of that index, and that adapter is about 30 bases long.
Okay.
You'd have to read through that to read into the index.
I thought that the sequence was going long enough to actually from what you have under number 1.
Yep.
That the sequence could go into the
It does not.
green part, but it doesn't.
Size-wise, you'll have a distribution of sizes. So if you had exactly the same size in all your fragments, you could do that. But that's not the way the library prep works. There's a sort of distribution of sizes. That's right. So we just re-anneal another primer that's much more closer to that index. All right. Let me think, is there anything else I wanted to cover? I think that was it. Okay. So I will confuse you momentarily. Is there something I can write on?
Move a whiteboard.
Yeah, can you grab that for me, man? That'd be all right. So I told you that there are 4 channels, and there are four fluorophores on each of our one on each of our nucleotides. That is the way it is for all the platforms except the NextSeq. The NextSeq is an evolved platform. It has a very interesting idea. So the NextSeq actually only has 2 colors, red and green, but we're able to deconvolute 4 bases from that. Does anybody know how we do that? Oh, because we've been doing this a long time. I'll show you how to do that. We're getting very sophisticated in the way we're doing. That's right. Yep. Yes, we do. That is exactly right. So what I can show you on here, I'll show you how that works. No. No, actually. Yeah, that's right. That's right. So that is the, and I want to mention it to you because ultimately that's the sequencing data you're going to be seeing in 5 days. So it's, I think, it's good for you to understand that. It'll look much different. And you only see green and red intensity. So how do we do that? And I'll just make note of it. Better? All right. What is this? Green, red. So in the NextSeq only, all the other platforms has 4 fluorophores. NextSeq has 2. So we have essentially a green intensity and a red intensity. And anything that shows up in green only is it's a T. I have to remember which one it is. It essentially is the green fluorophore is on our T base only, is on thymine only. If it shows up in red, it is a C. Cs have red base only. A base. Both. So you have equal red and equal green intensities on that. And G, a whole lot of nothing. So G is actually blank. So there is a couple things to consider when you look at that. And one of them is registration. So what happens if you have a string of G's at the front of your read? That's one of the reasons why we do 5 cycles of registration, because if you did 1 cycle, 25% of your reads would not show up because they have G's. They're blank. You wouldn't be able to see them. So you would have to have 5 G's in a row in order to lose a cluster. Probability speaking, that doesn't happen so often. So we're willing to take that. But if you have one other channel, one other base in those first 5, it'll find that cluster. It subsequently call any other blank space a G. So that's how we do that. But it's essentially gives you the ability, in a manufacturing standpoint, to minimize the amount of work that you put into making the kits and things like that. It's much more simpler to look at those kind of things. Calculation-wise, there's no crosstalks and things like that. So when we do 4-channel systems, there's overlap, there's bleed between them.
Both?
Yes.
Or like randomly, so some half A's of reds and half A's of greens?
All right. All right. So technically there are 4 fluorophores, all right, but there's only 2 colors. So the fluorophores that are on A are not the same fluorophores that are on T and C. In fact, they're twice as bright as the ones on T and C. Why? Because you're only binding half of each. So in order to get a normalized signal, you got to use a fluorophore that's actually twice as bright as the other one, because you only have half the molecules on that cluster, right? That makes sense? Let's say, I don't know what the fluorophores are, but
So your nucleotides, half of them are the red fluorophore, half of them are the green fluorophore.
on A, yes.
But they're twice the
That is right. So we in that incorporation cocktail, that is what we do. So it's all mixed together in there, right? So it doesn't, it's all based on the hybridization, right? The hydrogen bonding is what fluorophore gets incorporated, and in the A channel, in the cluster, half of them will be red, half of them will be green. And in G's have no fluorophore on them whatsoever.
This just simplifies the detection.
That is right. So we
Oh, I get it.
then we only have 2. Yeah, that's right. That's why your NextSeq is going to fly through this 300 cycles in one day. because it's taken half the images, and it's essentially recording things much, much quicker, right?
So you have issues with what it's called, like if you have the T that is lower and really close to G.
That is where the filtering comes in, that's right.
But like in theory, it's something that you probably
Yeah.
would be able to figure out before, you might not be able to figure out now, right?
So this is what we call chastity, what you're talking about. So chastity in the NextSeq is a little different than the other platforms, and it's ultimately a measurement of distance from a centroid of a cluster. So when I see this, I see Illumina is a genotyping company. We made essentially made genotyping projects to give us enough money that we can buy Solexa. That's what we did 7 years ago. So when I see this,
Second, we cannot explain it to each other, which is not a
We
good
can't
knowledge.
explain each other's best. So
all right,
if you've got an A molecule, let's say you've got 10 A molecules, let's keep it simple.
all right.
And so basically you're saying is 5 of them would have the red fluorophore attached to them, and 5 would have the green fluorophore attached to
Yep.
them. But how is, okay, but that, and then for T, if you had 10 molecules, 5 would have green, or
And that's it.
5 would have
10
green,
would have
and
10.
10 would have green.
10 would have green. But, so your incorporation, when you're adding fluorophores, it's 100% incorporation?
Yes. If it's not, that's a phasing value,
Right.
depending
And
if it's
how
forward
is
or
it-
right.
How is that molecule twice as bright?
Different fluorophore. So you get like Psi5 is way bright. No, there.
Molecule, but it's the same.
That's what I meant when I said technically there are 4 different fluorophores.
So I got it.
There's 2 channels, 4 fluorophores.
Fluorescence output, but
It is the same. It is the same, almost identically the same wavelength, but in terms of intensity, it's much brighter, twice as bright. This is something we worked out in chemistry. But back to chastity. So chastity is measurement of distance at this point. So when I look at this, I see AAs, ABs, BBs, right? I see genotyping calls when I see this plot. Illumina is very good at genotyping things and calling those very, very accurately. So that's NextSeq is sort of incorporating a lot of our array analysis methods and our information that we take from there, and this is one of them. So when you have, these are clusters, right? They're all intensities that are following here. So what we do is we find a majority of the population, right? And we draw this, we call a cluster on top of clusters, right? So you get this clustering algorithm that goes and finds this. But not everybody falls in here, right? It more looks, honestly, it doesn't look like this. It all looks like a shotgun blast, right? There's just dots all over the place, right? I mean, but they are centralized to certain areas, right? So when you're base calling things and you're looking at things, you're actually looking at these little these clusters, and you determine where the centroid is. Where's in this given run, everything's run specific. Where are the majority of these G channels falling, and what's the middle? And then when you have something that's sort of outside that, you start to measure distance. So this is D1, let's say. So when you want to find out what this cluster is, in the equation, chastity here is essentially D1 divided by D1 plus D2. And that needs to be greater than, I think it's 0.7 in NextSeq, give or take, it's the ratio. And what does that mean? So let's say this is D1. This is a value of 1. That's how far away it is. The second closest centroid is going to be D2. Let's say it's the G. And this is 1. So when you have 1 divided by 2, you get 0.5. This fails chastity. Make sense? Because that's absolutely right. You have absolutely no idea which one it falls in because it's equally distant from all the from 2 centroids. However, if this dude moves up here, then you can start to get maybe this is 0.2 or 0.1 or something like that. Say it's 0.1, and then this distance is 1. So 0.1 divided by 1 is sorry, that's the other way around. But you get to where I'm going at, right? It beats that ratio filter, and you get above. I think it's 0.7 at this point. That's the chastity cutoff. So your confidence or your reads and things like that, your bases won't even be called if they fail that criteria. That's the 20% loss in pass filter that you see, is because guys here are essentially falling in between centroids, where they're at, and your confidence of calling that base, you just don't have it, so it doesn't call it. Illumina's technology will be much more geared towards essentially not calling a base than calling an incorrect base. That is the way the software has been written. We don't want to call the wrong base. We'd rather call some ambiguous base and rely on coverage to compensate for that, right? That's why you do oversampling and things. So that's the idea. If you do that, things work very, very well. You have very high confidence in what you're calling in terms of variance and things.
So that's, for example, if there was some wind within the cluster.
Yes, for some reason
The
it's,
strand was a little bit off
yeah,
the center.
it can happen for a lot of bubbles and things like that. It moves intensities around, and this is on a per cycle basis. So the next cycle, that same cluster could be right in the middle, right? So it's, it just depends on what happens at that cycle as the machine is running. All right?
Yep.
Any questions about sequencing, cluster generation? That is it in a nutshell,
Yes, I do have a question.
pretty much.
So now, if I understood the way this worked, going back to the very, very beginning,
Okay.
so when you have
Step one, hybridization.
step one, yeah, when you basically have the glass slide and it's coated with your mix of primers or whatever you
Sure.
want to call them,
With some
and
polymer.
You put your library on that,
Mm-hmm.
okay, and the denatured library.
Yes.
So that they're good to attach based on blah blah blah. All right. So
And typically what we flow and so for the next seq, you're going to flow 2 picomolar.
okay.
2 picomoles of template are going to go on that flow cell. And that's literally related to the physical area of the lane
Yeah.
and keeping them spaced far enough apart that they don't bleed into each other and fail filter. That is what 2 picomolar does for you.
Now, and what you just said make my question even more
All right?
Why would you not just load the RNA?
Well, you need double-stranded stuff.
Why?
Um,
You don't.
you do,
But tech limerase might use RNA.
yeah. So all of the,
Reverse transcriptase as your first one.
so we do a DNA polymerase that's on the flow cell already, right? So the DNA polymerase is what's incorporating the fluorophore. And if you think doing a PCR reaction is hard, you should try to incorporate a giant fluorophore that's hanging off the end of your nucleotide, right? Not a lot of polymerases do that so well.
And I was gonna say, this is sounding like the,
I understood this so much. All right. So what if I'm going to need colors? Okay. All right, guys, you help me, okay? All right. So here is our little place, and we have that one of those primers,
Yep.
right? Now, if I come with my RNA, oh shoot! Wait a second. I need a tag. Can't I do a ligase on the RNA?
You can do RNA
It's
ligase.
RNA ligase, right? Okay. So I have RNA
Yep.
ligase.
It's in our small RNA kit.
You're recording, right? Okay. All right. So if I have my RNA
Yep.
with my tag, okay, added by RNA ligase, the index, the same thing that you do, okay?
You need RNA polymerase, yes, in there as well to do.
RNA ligase, okay?
All right.
And then I can do a reverse transcriptase.
Oh, okay.
Can I?
Sure.
Yeah, do an RNA template, DNA.
Okay, so I do an RNA transcriptase, green. RNA
You
transcriptase.
have to have one out of the other end that has the.
Okay, and now I need to be able to fold this over. Second cluster. Now I need to add something here, right? Okay. Well, can I not just do what you just did on the library, which is add the ligase and add another color. And add
Yeah,
the
maybe.
library.
Yeah, maybe.
You have to
so
ligate out
wait,
to
but
both
are you,
ends of your
do
RNA.
you have double-stranded RNA
I don't think
now?
there's
No, I have a DNA RNA hybrid.
So you need first strand
on both sides.
This is DNA because it's reverse transcriptase.
Okay, so use reverse transcriptase.
Reverse transcriptase, so this is single strand DNA with an RNA.
You'd have to have RNA with adapters ligate on both sides. I don't know if there's.
Won't ligase work on single strand?
Why would you want to do
The ligate
this
on both ends.
on the flow cell where you want everything to be as simple as possible,
Because I'm a
rather than
complicated.
in those two tubes over
Because
there?
I'm a complicated woman.
So Sanger has published a paper.
I mean, you can do
It does
RNA
actually.
sequencing, use direct RNA sequencing.
Yeah. So Sanger has actually published a paper that does something very similar to this, actually.
I knew
Where they do.
Sanger. He was a good man.
Yeah. The Sanger Institute has actually. I've read a paper like that. They do something like this.
And
Is
then
that they
you- and
actually
then
generate
you can do the
cDNA.
same thing that you guys are doing.
Yeah. The problem.
That's more complicated than 2 days of
You have to
library
look it up, but they do
construction.
have direct RNA sequencing where you kind of either mush or skip steps.
Yeah. And
But
put it on there. I've read
it ends
that.
up probably extorted. It's much more expensive and much more complicated.
Yeah. You're gonna see how fast your quality drops when you start doing things like that.
cost and complication thing, that it's more expensive and more time consuming. You have all this infrastructure that's set up for working with DNA, so all the enzymes, everything is cheaper. I think that than to go that way.
Considered it initially and decided it wasn't worth it. and they looked at
That's
it.
what.
So how do you put your indexes on those?
On what? On which one?
You're gonna
On the
have
ones
to
in your
set
flow
up
cell.
size.
Single strand?
So you can only do one. You can only do one sample. That's all that means. But
Oh, yeah.
because you can't identify them
You have
independently.
to separate them into separate molecules.
With a tube,
first.
pretty much. Yes, that's right.
When you
Right.
add
That's how you do that with a tube.
Right.
those
Yeah, good point.
oligos that you would have on the plate could have the index.
Yes, but they're going to essentially randomly ligate to all your different samples that are across
Yeah,
there,
you
things
could
like that.
only load
So you only
one
one
RNA
sample. Yep.
yeah.
But
Okay, you got
there are some development things that, if you look up the Sanger, you're probably talking about that Sanger paper or something.
Yeah, and they
It
have.
was many years ago they did that, but they did some things like that.
Yeah, I think
People do weird things like that.
Because I was talking about this at dinner the other night. That
I've seen a lot of weird
Clinical
things
Core Lab,
actually.
They have direct RNA sequencing for pathogen identification because it's just much quicker.
Yeah.
You don't have to do all this, but it's also much more expensive. So I'm just
And they
looking
have
to
certain
have
things
that.
they're looking for. I'm sure
Yeah,
they're
and they
looking
have
at
certain
very
things that they're
specific
looking
regions.
for.
They're not
Okay,
trying to get the
forget
whole
about it.
pathogen
Sure.
genome.
Yeah, no,
Okay, I just
it's the
have a
right
question,
idea.
too.
Yeah, sure.
So I must have something wrong because I drew like D1 would be the center of one to the center of the other, which would mean D1 and D2 would equal each other, and then it would always be 0.5. So obviously I have something wrong. So what does D1 exactly equal?
Distance to the closest centroid and distance to the second closest centroid.
Distance to the closest centroid, and then distance to the second closest centroid. So
That's right.
D1, and
Yeah.
that's D2. Okay.
And that is essentially determining signal purity, right? Because
Right,
if it doesn't
right.
falls in that sort of weird space, then.
And that's why if you load too much and
So you start to get all the bleeds into each other. That's right.
you're getting more and more.
So you start to loosen up your centroids and your things start to diverse
I
and
just had my drawing
yeah.
wrong, so.
No, it's very it's a very simple concept. We leverage it. It works very well. And it's the same thing we use to call alleles, right? AAs and BBs. It's the same exact idea. Photo bleaching? Yeah. So you do see that. So some of the machines now, not so big of a problem. So the NextSeq actually uses an LED light. It doesn't use a laser. So when you back off that intensity, you keep that photo bleaching down to a minimum. But you're only zapping it once at a location, right? So you take it and you image and then you move up to a different part of the flow cell and you take that image. So and then the fluorophore gets cut. So then you resynthesize you reincorporate the whole new fluorophore. You don't see any bleaching that way. And the reagents are actually separated from all the other machines, so there's no light or anything like that in there. They're all contained. But in the old days, we used to see that a lot. If you image the same tile or the same area over and over again, you'll see your intensity just drop and it's gone. Yeah, you'll bleach those out very easily.
I don't know how it's physically like edge effect. I guess
Yeah, we get some bleeds and flares and things like that. That's all just part of the optics, yeah.
that's never like
So we crop images. So the flow cell, the lane is much larger than the actual acquisition we take. So we put improvements into to alleviate that from coming into the data, actually.
but the LED is kind of like a huge improvement instead of like
Yeah, sure. There are, I mean, I think there is some weirdness that goes on, but we've seen that in-house, and we just make corrections to how we analyze things, ultimately to solve some of those problems. So one thing is, so there's two layers, right? So there's a top and a bottom surface, right? Because you graph the oligos, like again, in that tube, it's in three-dimensional space. So we actually image both the top and the bottom surface. It doubles our read number by doing that. And the bottom surface is 72 microns away from the top surface, so we know how big that flow cell is, right? We just drop it down and image that layer. We image the top first before we image the bottom, right? Because you don't want to pass the beam through the top, you'll bleach out all your clusters that way. So there's things like that that we do to keep things integrity of signal at its best. That's how we do that.